Let's jump right in. Think of life as the absolute ultimate architectural masterpiece. Seriously, imagine a towering skyscraper, but instead of being built from steel and glass, it's constructed entirely from carbon, nitrogen, and oxygen. Today, we're taking a journey of scale. We're going to start at the absolute foundation, the tiny molecular bricks that actually make up living tissue, and we'll build our way up from there. We'll see exactly how these molecules form cells, how those cells group into tissues, and how those tissues construct the incredibly complex physiological systems that keep organisms alive and kicking.
Now, this might sound like the setup to a really strange joke, but I want you to seriously think about this question for a second. What on earth connects a scurrying insect, a green leaf, and the organs that are literally purifying your blood right this very second? Well, the answer is scalable complexity. They are all absolutely brilliant, interconnected solutions built from the exact same fundamental molecular blueprint. They're just organized in radically different ways to solve the exact same problem: survival.
Okay, let's dive into this. Here is our roadmap. 1. The molecular foundation. 2. The cellular level. 3. Tissues and structures. 4. Anatomy of organisms. 5. Systems for survival. And finally, 6. Waste and maintenance.
First up, Part 1. The molecular foundation, the actual bricks of life.
So, how do scientists actually figure out the chemical makeup of living tissue? It's kind of wild, actually. They take living tissue and physically grind it up in a really harsh chemical called trichloroacetic acid. When they strain the sludgy mixture, it naturally separates into two distinct parts. The liquid that passes through the filter is called the acid-soluble pool, and this gives us a pretty good representation of the cell's cytoplasm. But the stuff that gets caught, the retentate, that is the acid-insoluble fraction. And this is where we find the real heavy hitters, the biomacromolecules. These are the large, super complex structures doing all the heavy lifting in living things.
One of the absolute most important biomacromolecules found in that acid-insoluble fraction is protein. But a protein isn't just some boring, repetitive chain of the exact same link like a homopolymer. No, it's what we call a heteropolymer. It's made of completely different types of monomers, specifically an array of different amino acids. Now, this is absolutely crucial for your own nutrition. Your body actually can't synthesize all the different amino acids it needs to build its proteins. Certain ones are essential, which literally means they must be supplied through your diet. So the dietary proteins you eat are the literal building blocks you consume to maintain your own molecular foundation.
All right, moving up a scale. Part 2, the cellular level. Think of these as the rooms of the house.
When a cell divides, it goes through several distinct phases, and this brilliantly illustrates the sheer mechanical precision of life down at the cellular level. Take anaphase, for example. During anaphase, the centromere, which is that central tie holding two identical chromatids together, splits completely simultaneously. Snap! It's exactly like a starting gun going off at a race. The chromatids are then physically pulled away from each other toward opposite poles of the cell. This whole perfectly choreographed dance ensures that when the cell finally divides right down the middle, each new cell gets an exact identical copy of the genetic blueprint.
Let's keep zooming out. Part 3. Tissues and structures. This is where we start building the walls.
A fantastic example of this structural building is epithelial tissue. Let's look at simple epithelium first. It's just a delicate single layer of cells. That makes it absolutely perfect for acting as a lining for body cavities, ducts, and tubes where things need to pass through. But then you have compound epithelium. This type consists of 2 or more cell layers stacked up. Because it's significantly thicker, its main job isn't lining a delicate internal tube at all. Its job is hardcore protection. In fact, if you just look down at your own arm right now, you are literally looking at compound epithelium. Your skin is specifically designed this way to protect all the vital systems underneath.
But even within that delicate single layer of simple epithelium, the rule of biology holds true. Shape dictates function. Squamous cells, for instance, are super flattened with irregular boundaries. Because they're so unbelievably thin, they are perfect for forming diffusion boundaries. Think about the air sacs in your lungs, where oxygen needs to easily pass right through the tissue. Cuboidal cells, on the other hand, are shaped like little cubes and are found in the tubular parts of kidney nephrons. They completely specialize in secretion and absorption. The specific architecture of the tissue perfectly matches its job in the body's internal plumbing.
All right, zooming out again, Part 4, Anatomy of Organisms, the complete structure.
Now, what's really interesting about this slide is that despite its seemingly simple appearance, a typical leaf is highly organized for photosynthesis. It actually consists of 3 main parts. First, there's the leaf base, which attaches it directly to the stem. Then there's the petiole, which is the stalk that holds the leaf blade out to catch the light. And finally, the lamina, the wide flat blade itself, where the actual magic of photosynthesis happens. It's a structure perfectly evolved to act as a highly efficient biological solar panel.
Now, contrast that plant leaf with the complex anatomy of an insect, like a cockroach. Its body is highly segmented and divided into 3 primary regions: the head, the thorax, and a 10-segment abdomen. The thorax alone is an absolute marvel of biological engineering. It consists of 3 segments that bear 3 pairs of walking legs and 2 pairs of wings. And inside, it houses an incredibly well-developed alimentary canal. Basically a full digestive system complete with a crop for storing food and a gizzard for grinding it all up. From a simple leaf to a complex insect, we see how tissues combine to create highly specialized survival machines.
Which brings us perfectly to Part 5, Systems for Survival. Basically, the plumbing and ventilation.
It turns out an animal's habitat and its level of structural organization completely dictate how it breathes. Simple organisms, like sponges and flatworms, they don't need complex lungs at all. They just exchange oxygen and carbon dioxide by simple diffusion across their entire body surface. Insects, however, use a fascinating network of internal tracheal tubes to transport air directly into their tissues. Aquatic creatures, like mollusks, they rely on vascularized gills to pull oxygen straight out of the water. Meanwhile, terrestrial vertebrates, like reptiles, birds, and us mammals, utilize highly specialized vascularized bags called lungs. Form always follows environment. But survival isn't just about breathing, right? It's also about resource management. And plants show us a brilliant example of this when they are starving for minerals. If a plant lacks what we call mobile elements, like nitrogen or potassium, it will literally cannibalize its older dying leaves to move those nutrients up to the younger growing parts. So naturally, you'll see the deficiency in the old leaves first. But if the plant lacks immobile elements like sulfur or calcium, which are permanently locked into the structural tissue and just can't be moved around, the older leaves stay totally green, and the new young leaves are the ones that show the starvation first. The plant's internal logistics completely dictate where the starvation shows up.
And finally, Part 6, Waste and Maintenance, System Upkeep.
Here's the thing about processing proteins. It creates nitrogenous waste. And evolution is designed 3 major ways to get rid of it, based entirely on how much water is available. First, ammonotelic animals, like many aquatic fishes, just excrete straight ammonia. It's highly toxic and requires massive amounts of water to flush out, which is totally fine if you literally live in a lake. Second, ureotelic animals, like us mammals, convert that ammonia into much less toxic urea, which we can excrete with only a moderate loss of water. Finally, uricotelic animals, think birds and reptiles, they need to conserve maximum water. So they excrete uric acid as a thick paste or pellet. You know that white bird dropping on your car windshield? Yep, that is literal evolutionary water conservation in action.
Since we mammals rely so heavily on our kidneys to filter that urea out of our blood, it raises a pretty terrifying but very real medical question. What actually happens when this complex ureotelic system in a human experiences acute failure? What do we do when the biological plumbing just stops working?
Well, when the kidneys fail, waste builds up to lethal levels super fast. While we do have interventions like dialysis, the absolute ultimate medical correction for acute renal failure is a kidney transplantation. So the crucial point here is this. When doctors perform a transplant, they don't just want any kidney. They strongly prefer a functioning kidney from a donor who is a close relative. But why? Because we have to outsmart the body's own fierce defense mechanisms. A kidney from a close relative massively minimizes the chances that the host's immune system will recognize the new organ as a foreign invader and reject it. We are essentially using biological similarity to heal biological failure.
Wow, okay, we've covered a ton of ground today. We started all the way down at the trichloroacetic acid analysis of tiny, microscopic molecular components. We literally watched chromosomes split in a single cell. We examined the protective tissues of our own skin, the segmented body of a cockroach, the breathing tubes of insects, and finally, the incredible complexities of human kidney transplantation. From the absolute micro to the stunningly macro, life is truly a masterclass in scalable architecture.
So I want to leave you with this final thought to ponder. If all of this incredible, wild diversity, the gills of a fish, the leaves of a plant, the ureothelic system of a mammal, is constructed from the exact same basic molecular bricks, what other unbelievable architectures might life be capable of building in the future, or perhaps elsewhere in the universe? Keep questioning, keep looking really closely at the structures around you, and thanks so much for exploring with me today.